Hi, my name is Basak Selcuk. I'm a graduate student at Oregon Health and Science University. Today I'm going to talk about iterative graph perturbation to identify high-impact nodes with application to genetic regulation of onset of puberty. Every day, more and more complex phenomena are explained by networks. With the real-life complex interactions such as seen in biology, with large number of elements, the networks become complex with specific characteristics, such as cluster formations and hub node generations. Figure 1 shows a sample view of such a complex network. There are clusters and hubs with high connection values. Not all the elements of a complex network have the same level of importance when the network function is considered. There are nodes that affect the network function when eliminated. Here, we have developed an iterative graph perturbation algorithm to identify nodes with the greatest impact on a given graph or network model. In our research, we try to understand the initiation of puberty network. Puberty is the developmental phase during which profound hormonal, physical, behavioral, and psychological changes occur. Researchers have proposed that there is a core gene network responsible for the timing of puberty. Figure 2 depicts a summary of research in genetics of female puberty. In humans, mutations in about 30 genes were found associated with early or absent puberty. Genome-wide association studies identified more than 300 genetic loci associated with the age when girls first start menstruating. This is a clear indication that puberty development is a polygenetic process. The whole initiation of puberty network is complex, with clusters of different functional genes working together, and it is still not completely understood. The goal of the impact node finder algorithm is to find a subset of the nodes whose removal causes the biggest change in the function of the network. These should be the nodes that control the network's function. We developed a heuristic beam search algorithm in which the beam is competing subsets of nodes. Subsets in the beam are iteratively added to. Using a beam search helps us avoid local minimum. It also allows us to return multiple candidates back for further exploration. We used regularized partial correlations to calculate edge weights when creating the networks. There are two levels of pruning. For node-based pruning, we use the union of node degree and node strength metrics. For network comparison metrics, we looked at different choices. Here, I am going to only include Jaccard index network metric that measures similarity between network clusters. We check final criteria to decide confounders to remove from the beam and add to the results list. Figure 3 is the pseudocode of our algorithm, which I am going to explain more with an example next. Figure 4 shows how network comparison works. G0 on the left is the initial network, and G114 on the right is the new network constructed after taking node 14 out. The different colored node groups are the clusters. The nodes with different colors in two networks are the ones changing clusters. These are the nodes that affect the similarity of the two networks measured with Jaccard index. Figure 5 serves as an example of how the INF algorithm works. We start by constructing an initial network that includes all nodes. At the first iteration of the beam search algorithm, by using the node characteristics, we find the first set of potential impact nodes. These are the first confounders of the beam search. By eliminating one node from the node list at a time and recalculating the edge weights, we construct new networks. Next, if no networks meet any of the final criteria, we calculate the change in the networks from the initial network according to a chosen network comparison metric. We choose the most different networks according to a predetermined beam width, and by only considering these networks, we start the second iteration of the algorithm. To test our INF algorithm with controlled and less noisy data, we created simulated gene expression datasets using the Java app Sintran with random seeding. We looked at the results to find transcriptionally active nodes in a gene expression network, which includes both active and background nodes. We generated 100 datasets with 10 active nodes each. Figure 6 is the Wayland plot of median number of active nodes found per iteration. Each iteration corresponds to an addition of a gene to be eliminated, as mentioned before. Figure 6a is the active nodes found by only using initial networks. Figure 6b, which depicts a steady increase of active nodes found, is the Wayland plot of results when using INF algorithm. Our results show that our method could separate transcriptionally active nodes among background nodes better compared to the initial networks. For our biological results, we investigated genes found from the first 30 iterations. Our results are based across all contenders in the beam. We looked at functional gene groups that mapped to enrich our database with an adjusted p-value of less than 0.05. We found genes that matched previously known functional puberty gene groups, as well as new candidate genes. Figure 7 shows our results for gene ontology and transcription factors. The y-axis shows negative p-values, so the values on the top are most significant. These previously recognized functional gene groups that we confirmed with INF algorithm shows that the newly found genes are good candidates as biological targets for further experimentation. In conclusion, our simulated data results show that our algorithm can differentiate between transcriptionally active nodes and background nodes in a network. Biological data results show enrichment in gene groups and identified new genes in previously known puberty-related pathways and mechanisms. Our results align with previous findings from the Lomnitzy lab, where they demonstrated the central role of two families of epigenetic modifiers, the PCG and the T-Rex group of epigenetic activators, in the control of reproductive development. This is the end of my presentation. Thank you.